Well, hello everyone, and welcome to the 15th annual event in the Clinical Diagnostics and Research Virtual Event Series brought to you by LabRoots. Today's webinar, Embracing the Digital Lab Movement to Improve Clinical Diagnostics and Research, is sponsored by eLabNext. For more information on our sponsor, click their logo found on the left of your screen. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's presentation. Now, on your screen are several interactive windows, and we encourage you to engage with them during the presentation. You can speak with other attendees in the Chat with Your Peers window, or you can submit questions for our speakers in the question submission window found on the bottom right. I would now like to introduce our speakers. We have Zareh Zurabyan, Twan Spenkelink, and Marisa Dunigan. To view our speakers' complete biographies, click their names found on the left. I will now hand it over to our speakers to present Embracing the Digital Lab Movement to Improve Clinical Diagnostics and Research. Over to you, Zareh.
Thank you. Thank you very much, Israel. Thanks to LabRoots for hosting us. Very excited to give this presentation to you all. And big thanks to Marisa Dunigan from MSKCC for joining us, and of course, Twan from Europe. So let's get started. Let me give you a quick breakdown of the agenda as to what to expect for the remaining webinar. So I will talk to you about current challenges in clinical diagnostics and research and what we have noticed. What is a digital lab platform to begin with, and how that can help the labs to optimize their R&D in the clinical space. We'll have a special demo of a product that will show you how to manage your samples, your reports of a clinical diagnostics lab. Then we'll pass it on over to Marisa from Memorial Sloan Kettering Cancer Center that will talk about their digitization journey. Then in the end, we'll open up for a Q&A session. Very much looking forward to that. So how digital disorganization undermines lab success. We will be talking about the key challenges that we have noticed over the course of the years, and hopefully we can share some insight on how to overcome that. So challenges in clinical diagnostics and research that we have noticed starts from many different roots. And in the end, it all affects something that is the most important thing in clinical diagnostics, which we will see in a little bit. So one thing that we have noticed is data volume management and standardization is lacking in many labs. So if you're working with flow cytometers, HPLCs, mass specs, PCR data, this is terabytes of information that live on many different computers, on many different servers, creating deep data lakes, which then becomes very problematic to manage, centralize, and then analyze. Sample mismanagement is another huge problem in clinical research, whether samples are not being labeled or they are mislabeled. There is no chain of custody as to how these things have moved around the lab. There's no audit trail for people to know what has happened to it. And then, of course, just loss of samples because of things being moved around and so on. And this can cost thousands of dollars, if not millions, for a clinical project. Then, of course, we know that in clinical research, there's a lot of collaboration that needs to happen. If it's in healthcare, there's a lot of collaboration with other departments in the core facilities. And we have noticed that without having a digitized space, there's a lot of interdepartmental miscommunication and loss of data. Regulatory compliance and quality control, of course, this is a key component of clinical diagnostics and research. Believe it or not, many labs are still going through a lot of paper lab notebooks and looking through page by page and trying to track things down. And this becomes a huge time commitment, which then slows down a lot of the work that scientists are doing in the lab. which then really causes a lot of expenses and costs that slows down the projects and runs through grant money or any investment money for any laboratory. This in turn ends up impacting the time in which projects are being completed. And of course, this essentially means that the therapeutics that we are hoping for to get out there to help the patients is taking much longer, costing human life. So essentially, all of these things is impacting patient safety one way or another. Oftentimes, we only look at the low-hanging fruit, the immediate kind of thing that we're trying to do. For example, hey, I want to see where my samples are. And of course, that's a valid thing to want in a lab and in a digitized lab. But it's important to see the cascading effect of that and how that can impact patient safety long-term if the lab is not digitized. So to take this further, let's talk about the journey of lab digitization. It's important to look at these kinds of solutions that provide digitization solutions in a way that will help you to know exactly where you start and where you will end up. And this is something that will take some time, right? So when we look at manual data and sample management, we realize that this is very timely and costly. Data reporting can be strenuous, analytics can take a very long time, and lab operations can fail. And in the short term, we try to transition to a digital sample and data management and lab operations. This transition can take a very short period of time if you have all of your ducks in a row and you have the right consultation throughout that process. What this ends up happening is it creates the result of organized lab and data. And what this essentially does is it creates efficient digital and business strategy for your lab. Yes, also business strategy in case if you are a core facility, in case if you are just an R&D lab that's not going into clinical. It's important to look at your research in a way that will do a lot of cost saving for you to use that money to invest in the right instrumentation and in the right people to come in and help you to elevate the research that you're doing in the lab. And lastly, the long-term impact of a digitized lab is creating a sustainable and future lab ecosystem that is scalable and is going to impact your business operations at large. And we're not using the word sustainable as a buzzword here. This is a true statement in that when you create an ecosystem, a digital ecosystem that's sustainable, this means that if you leave the lab, in case if the research gets pivoted into another topic or another department, all of that work that you have created, all of the integrations, all of the digitizations that you've done now becomes usable and scalable regardless of any changes that happens in the lab. So what does a digitization journey stand on? What are the pillars of success? The end goal of a digitization journey is to create data integrity and data connectivity while maintaining digital health. And the pillars of this are accessibility. And this is not just about accessing data remotely from home, which has become a very hot topic since the pandemic, but this is about accessing very specific sets of data. So we were talking about deep data lakes, we're talking about terabytes of files, but what about all of the reports, the essays that are being completed? If you're a lab of 30 people and each one has their own projects that they are completing, it's important to have standardized and indexed data that will allow you through just a couple of clicks be able to access that information. Then there is data analysis. Whether you are using GraphPad, worksheets, Power BI, or any of these other systems that you are pushing the data into to do your data analytics, or if you have a data scientist on staff that will be parsing through all of that information, it's important to be able to do that in a structured way. So having a digital lab platform that can provide that is very important. And there's other ways to actually automate that outside of just the front-end UI through leveraging the API and SDK, which I'll talk about in a little bit. Security and compliance, another huge component of lab digitization, which I'll talk about shortly in another slide. Instrument monitoring and connectivity. This can mean anything from having the temperature monitoring of your freezers, of your biobanks, being connected to your digital lab platform, all the way to being able to control your Hamilton robots and your TCAN robots directly from a digital lab platform, or being able to automatically sync files from your flow cytometer directly into your digital lab platform. And lastly, the scalability is extremely important, because historically, digital tools were created for very niche and very specific kind of workflows within the lab. And these labs, when they started to scale up, they were not able to scale up with that system, forcing organizations to buy 7 or 8 different software, causing decentralization of your data. So here it's important for everyone to note that having a system that can leverage its API and SDK stands for Software Development Kit, that allows you to integrate third-party solutions, or being able to integrate into your, let's say, hospital data centers, or whatever the case is, to be able to have that connectivity, which then will allow you to scale the system as the samples are scaling in the lab. So hopefully this slide here puts a smile on everybody's faces. So, this is what we want, right? In the lab, we want to be happy. We want everything to be digitized, and we want that digital solution to actually help us physically in the lab. And oftentimes what happens is that we end up being the person on the bottom right. I'm sure many of you have seen this maybe in your lab, maybe in previous labs, or in other people's labs. I want to put this out there, is that do not feel bad. This is a common problem across the globe, in academia, in pharma, and in healthcare, and in small biotech. And this is not because scientists are not good at what they do. It is because we simply haven't had the tools available for us to create a structured and organized sample management, data management, and reporting in the laboratory. And what ends up happening with a system like that is you end up sacrificing the most important element in clinical research, which is patient safety. So it is important to overcome those challenges in order for you to unlock the potential of these laboratories to invest in their discovery, invest in their innovation, and scale in that process. So that brings us to another topic here, which is security and compliance. And it's important for us to note that if you are looking to manage clinical data, patient data, it's important for you to abide by all of these. This is HIPAA compliance. This is GDPR compliance. This is for EU in particular. GXP in many cases is required. ISO certification, VPAT compliance, CLIA compliance. I mean, you guys have seen many of these acronyms thrown around. And having ISO certification is probably the most important one whenever choosing a digital lab platform, because this is the highest level of security and compliance that you can get on a data management system that is cloud-based. And in order for you to be CLIA compliant, this means that you have to be on a private cloud or an on-premise installation, and you cannot use cloud solution by default. That is not allowed. And also you need to ensure that the company itself is ISO certified and that they're not just using ISO certified data centers. So with that in mind, let's look at this questionnaire. I'll give you guys just a couple of minutes to fill this out. So if you can put in here which compliance is the most important to you and to your lab. 21 CFR Part 11, HIPAA, GDPR, CLIA, or all of the above. Give you just a couple of seconds, and then I'll move on to the next slide. All right, let's move on to the next slide. So what happens with these labs that decentralize their data by using 7 or 8 different software, or maybe they're not using any software and they're using they're using paper lab notebooks, or maybe Word documents or EndNote, Evernote, and kind of sharing the data in those ways. What ends up happening is you disconnect your data sources. You create silos. And what this ends up causing is a lot of inefficiency. You lose convenience, and you lose the quality of reproducibility of your results. There are so many labs across so many institutions that are getting sued left and right these days because they're not being able to reproduce the results. And that's because the data is disconnected from the actual research that was done. Then if you are using many different software that provide very good solutions for that particular thing, this creates a problem as well, because you end up having a one-dimensional solution. So maybe you're using a homebrew Excel or a LIMS system. Maybe you are using one system that is specifically for molecular biology, another one that's specifically for the drug discovery component of things. Again, the communication between those departments and those people creates a lot of issues. And that only provides one piece of the puzzle versus the whole puzzle. So it's important for you to look at the whole puzzle and not just the pieces of it. And the current available tools out there are one-dimensional and are not future-ready, which then causes lack of connectivity, scalability, and they're too rigid in their technical capabilities to help you to maximize what you need. So what we suggest to you, to the listeners, is to think about what can you do to become future-proof in the digital solutions that you are choosing. And to help us to understand that better, a little bit of a history lesson here. So let's think about it. So I know I've used these kinds of phones before, and this was established in 1861, first time, the cable landline, and physical notebooks and keeping track of your samples physically is as old as science. Of course, we've been taking notes forever, and it's something that's in our DNA. And maybe this is something that will always be part of our day-to-day operations in the lab. So then we move into the 1990s, which is the transition from analog to digital. And we had mobile phones, which were established in 1996. And we had the very first electronic lab notebooks and LIMS and so on, which became more prominent in 1994. But the problem with those is that, again, they were not scalable. They were just in-house made systems that were not able to scale. So what happened is, in the recent history, we have the smartphone, which was established in 2007. So you no longer need to carry a calculator with you. That's within the smartphone. You no longer need a GPS. That's in your smartphone. And with eLabNext in particular, but giving you advice on how you can choose the kind of digital lab platform, is thinking about a system like a smartphone, like an ecosystem, which will be able to provide you with that ecosystem where you can go ahead and integrate third-party apps, third-party add-ons into your digital space to expand the capabilities for your organization. So with that in mind, some challenges that we have encountered in the data management process that I want to go over. I won't read through all of these, but I do want to have this here that you guys can go back to and look through all of these questions. But I do welcome all of the listeners to not only think about the immediate needs of your digital solution that you are choosing, but also to think about the long term. So for example, we are noticing now that AI is becoming a huge part of our lab operations. And maybe right now you are not using AI tools, but 2 years from now, 3 years from now, it's highly likely that you will be using AI tools. So it is very important for you to start structuring your data of your biobanks, of your samples, and all of your reports and assays that you're creating now, because then you will be able to digest that data. Data scientists will have all these amazing tools available at their fingertips that will be able to utilize with structured data. If the data is not structured, it is a waste of time for them to go back into all of your different data silos to look through that. So another thing to think about from all of these questions that you see, which might not seem obvious, but I do welcome you guys to think about, besides the short-term and long-term goals, is, for example, if you know how all of your samples are managed in your freezers, and you are doing less of that opening and closing of the door of those freezers. And if you are a large institution that has hundreds of these freezers, well, guess what? That is a lot of energy consumption that the building is suffering from. And you will notice hundreds and thousands of dollars being saved from simply knowing where your samples are and being able to access them in a smart way, and not through a trial and error by opening and closing and so on. So I just wanted to bring that example in particular here also, because people don't think about those things. We quickly kind of jump into thinking about the more grandiose kind of goals that we have for our labs, but there's a lot of these trivial cascading effects that can really optimize everything that you in the lab for you to consider during this digitization journey. So before I pass it on to my colleague, we have another question for you all. I'll give you guys a couple of minutes to fill this out. So now you've kind of looked through some of these hot topics. Which features of a digital lab platform like ELN and LIMS are most important for your research? Is it clinical data reporting? Is it clinical sample management? Or it is colleague collaboration? Give you guys 10, 15 seconds to fill this out before I pass it on to my awesome colleague, Twan, who is joining us from Europe, that will talk about the future of clinical diagnostics and research within digital labs, and run through a demo of what this could look like digitally. Take it away, Twan.
Thanks for the great introduction, Zareh. Really appreciate it. Before I will share my screen, today we will take you onto a journey on what your management, your digital lab could look like in terms of a clinical trial workflow. Within this journey, a couple of key points, key aspects are really important, of course. We are talking about compliancy, GXP compliancy, CLIA compliancy, about patient safety, collaboration, but please keep in mind that this journey is just one of the examples of a flexible system that we can utilize. Your journey can look similar, but can also look different in a couple of aspects, or totally different. Please keep in mind that we are really flexible, especially within the digital lab platform that we are utilizing today, the eLabNext digital lab platform. I will share the screen where I have already set up a digital lab platform environment, including a clinical trial workflow that is already included inside of this environment. We will go over the key aspects here and share a couple of elements that are really important for a digital lab platform to have. In this case, one of the key important aspects that we will go into is the interconnectivity of the data. We will not only have experimental data or research data, but also, of course, sample data, storage locations, and perhaps lab equipment that we would like to manage. For me, when I worked in contract research, I was also working on these kinds of projects and digitizing the lab. For me, that was a really important aspect. Having everything interconnected makes sure that we can really easily go into a centralized system and we won't have to go into separate systems to see certain data. One of the other key aspects here is, of course, making sure that we have boundaries for people and make sure that we specify if someone is, for example, qualified or cleared to access patient data, or qualified and cleared to make sure that data can be removed or not. This is also one of the aspects we can really easily configure within the workflow that we have set up here. On my end, we have a couple of group members implemented right here. The group members have their user roles already set up here, and the user roles really define what these people can do and what they cannot do. So again, like Z was also saying, in terms of compliance and in terms of having these boundaries of people, if they are cleared or not to access certain data. Again, this is all fully customizable. Having the permissions here in place, we have a customizable system, and for almost every action within the system that we have in the digital lab platform of eLabNext, we have a permission setting here, giving you a lot of control and a lot of flexibility over what people can see, what they can do, what they can remove, what they can modify, to make sure that everyone that has the right training also can perform certain actions within the system. Furthermore, as a clinical coordinator, or for example, as a QC manager, there are other compliancy settings available as well that we could really easily set within the overall system of the digital lab platform, making sure that all of the compliancy is checked. For example, if we are working with digital signatures for the GXP compliancy, it's all covered right here. This will take away a lot of pain points that you might currently have with the current way of working. If you are looking at this from a journey within a clinical trial workflow, we usually start off with a study, or for example, with a cohort of different kinds of patients that we would like to do our study on. In my case, I will start off with the cohort that we are using, so the patient data. Here, the key aspects which are really important are the patient safety, making sure that their data is safe, and also make sure that we can associate any sampling that has been done on the patients. In this case, I will move on into our flexible inventory system that we have within the digital lab platform. This is a flexible setup of anything that we want to store digitally within our digital lab platform. We got 3 panels right here. 1 panel for storage locations. Anything can be a storage location. If it's a freezer, if it's a storage location that we want to store chemicals in, if it's a safety cabinet, it doesn't really matter. If you want to keep track of that storage location in a digital lab platform, we are able to do so here. The key topic here is to basically create a digital copy of all of the storage locations that you have within your facility. In the middle panel, we can see that we have a grid layout of different kinds of samples that are currently placed within this sample box. This is working towards the point Z was mentioning about the way samples are managed within, for example, a -80 freezer. We have a digital copy of our setup right here. We don't have to open up the freezer to see what's in there. There will be less ice forming and less energy consumption on that freezer throughout the years and throughout the usage. On the right side of the panel right here, we can see directly all of the sample information of the samples that are placed within here. Looking at our cohort, we also have some patient data stored in here. Directly within one overview, we can see what kind of patients and patient data is stored in here, and we can look at the specific patient data. In this case, we have a patient with a anonymized patient ID of the age 79, And we can go a little bit deeper into this patient to make sure that we can view all of this patient sample information right here. In this system, we will work with a couple of structures that will make it easy for you to manage this kind of data and to make sure that it's all fully compliant, it's traceable, and that years later, if we are naturally growing our information base right here within a digital lab platform, that we can easily find back our entities and samples. If you are going a little bit deeper into the configuration of this, we can really easily, if we are clear to do so, configure new storage units within the system and also create new sample types within the system. The sample types are used to generate the templates that are being defined, that define what kind of information is being placed on each kind of sample. Looking specifically at the storage units, we have a couple of storage units available right here, but you can really easily add new storage units right here to make sure that our digital lab is set up in a good way. To make life easier, we have a couple of templates ready that will make it really easy for you to set this up. But please be rest assured that we always have the custom option as well if you want to fully customize the options that are available. Having this easy setup and this easy configuration of the storage units will make sure that it will take you more or less 10 to 15 minutes to set it up. And if you have more storage locations that are looking alike, we can really easily copy the storage units as well. So you can just do a one setup of a freezer and copy them over to make sure that we have similar setups ready in the blink of an eye. If we are going a little bit deeper into the samples that we had a look at earlier as well, we have this box with the sampling that has been done of a couple of patients. In this case, we can see that we have 3 types of samples ready. We have the blood sample, the tumor sample, and the urine samples that have been taken from these patients. We can also have a little look in the patient, the sample information that is available here and see the blood specifications. So all of the information that is available on this blood sample. We also have, for traceability, QR codes implemented. And the best thing here, of course, as a coordinator or as a QC manager, I want to make sure that everything that has been happening with this sample is, of course, also fully audit trailed. Anything that has been happening with the sample is displayed right here. And we have a full traceability with timestamps, as well as user-based stamps right here that we can see what kind of actions have been performed. In terms of sample overview, we have all of the data here, and the data that is linked to a certain sample of a certain type, in this case blood, is also fully customizable. With this, we will really go into the power of the system, actually. When I want to set up my sample database, I want to make sure that years later down the road, or maybe even next week, I want to quickly be able to filter on these samples and to make sure that if I'm interested in a specific certain set of samples, that I can really easily search for them. This is really easily done within this system, as we have customizable sample types that allow you to make sure that anything that you want to know about a specific sample can be defined within the sample type. With different samples, different sample types and different sample fields are available, which makes it really customizable for everyone that you have within the that you have clear to do so within the lab. As mentioned in the beginning, one of the key aspects right here, which is really important, is the interconnectivity of these different modules. Now that we have touched based upon these different structures that we have within the inventory, so it's the storage locations, the samples, and the sample types, we can also link them to our research or to our clinical trial flows. In my case, I already prepared a clinical trial that we are doing at the moment. So we got the cohort of the different patients right here. And we can actually have a look on what kind of clinical trials are currently running on this patient. So this will give us a lot of information about the current stage this patient is in. We can see that this has been linked to the clinical trial, and we can actually have a look at how this clinical trial is looking like by simply choosing the stage 1 that we are currently going into, and we will be directly going into the journal part of the system. This will allow you to flexibly go into the clinical trial stages of the study in which these kinds of stages are currently stored in. Again, full traceability and full connectivity to the inventory that we already touched base upon. In this case, we are collecting all of our information in this clinical trial in the structure of the digital lab platform, which makes it really easy to make sure that we have a overview of all of the data that belongs to this experiment and to this study. We have our collaborators in here, and we have a overview of all of the information. In this case, there are a couple of steps that we still need to perform. And as a coordinator, I want to make sure that all of these steps are clearly laid out. So in here, we got the patients also linked directly to the experiment. We got the blood samples linked, and we see that we still need to link our tumor samples. In this case, I will quickly link our tumor samples by going into the sample box, and I will select our tumor samples that we also are utilizing within this clinical trial. Now, within a few clicks, I just linked fully our tumor samples directly to our clinical trial and make sure that they are fully interconnected. In this case, we also connected a protocol or a standard operating procedure. So everyone that comes into this experiment or into this clinical trial knows that I, as a user, have done the sampling based on this protocol. If maybe later down the road something has proven to be going wrong with this experiment or clinical trial, we have a full traceability of what has been happening, including all of the steps and all of the samples that have been used. In this case, we also even have the equipment that has been used also linked to our equipment. Perhaps the equipment has been broken for a while, but we haven't seen it yet in the results. But having this fully traceable solution will make sure that we always have a full tracking, a full backlog of what has been happening within this clinical trial. Additionally, we always also can go into the sign and witness sign completion workflow as well. If the sampling has been done and this stage 1 has been finished, we can sign off the experiment to make sure that it will still be locked within the system, but that it cannot be changed anymore. As well, again, very important for compliancy. Down the road, the real power of the system comes into place when we are going into the connectivity again, but also if we want to really go into the power of the system, where later on we want to search for information and we want to see the interconnectivity of the information that we have added into the system. There are multiple ways of gathering that information. One very important way of gathering information about, for example, your patients is perhaps seeing what kind of experiments have been done with them, but also what kind of samples, etc., have been generated. In this case, we also have very advanced visualization options available. You can already see that we here have a patient of which certain sampling has been done. One very important aspect of the system is as well that you can see that some of these samples have already been archived. Perhaps they have been used up or have been fully analyzed. Within a digital lab platform, it's very important that anything that has been archived is still traceable. So here we can see while the sample has been archived, we can still see the full connection and the full interconnectivity of everything that has been happening. Also, when we want to have a full backlog and a full tracking of everything that has been happening within a certain study, we can also really easily get it out. This is really the power of a digital lab platform, where we can really easily go into a create a report actually of everything that has been generated within a certain study. In this case, I want to know all of the samples and everything that has been generated within our new drug study that we are doing within the digital lab platform as the clinical trial. This will give me an immediate result in here. So this is give me a full report of everything that has been generated, which will again mark the importance of having everything tracked and having everything implemented within the system. Before I'm handing over to Marisa from MSK, I will stop sharing my screen and go into a poll question for you to answer, where we give you a couple of minutes to answer. In this case, on a scale of 1 to 10, how likely are you to consider implementing a digital lab platform, such as an ELM or LIMS solution, into your lab to track your clinical research? So either a 1, no thanks, I'm not interested. A 5, this seems interesting, or a 10, I'm very interested in implementing such a digital solution. I'll give you a couple of seconds to think about this and move on to the next slide to move on to MSK and Marisa.
All right. Thank you all so much. I'm really excited to be here and tell you about the digital lab transformation that we've done here at MSK, specifically in our core facilities. So I'm going to start with just kind of explaining what a core facility is in case anybody is not super familiar. A core facility, or also known as a shared resource center, is a centralized resource within a research institution that provides access to specialized equipment, technologies, and expert services. So, our facilities are really designed to support our scientific and clinical research here at MSK by offering services that would be too costly or too complex for somebody to have in their own individual lab. So, there's 4 main types of core facilities that we have, and they fall under basic research, translational or clinical research, drug discovery, and infrastructure. So, really, we have inside of MSK, the core facilities are really here to serve a function to really help advance our research for MSK as a whole. So just to go over some of the fancy instruments that we have in some of our core facilities. So this is our integrated genomics operation core facility, and this is one of their next generation sequencers. So this, really we can start with some tissue samples, perform genomic sequencing to understand what mutations are present and what's driving the cancer in our patients. We have our nuclear magnetic resonance core facility that performs NMR spectroscopy. This can be used to characterize small molecules, which can help identify potential therapeutic targets to assist in the development of new cancer treatments. We have in our structural biology core facility, we have a cryogenic electron microscopy machine, which you can see a picture of right there. Um, a cryo-EM microscope can help determine the three-dimensional structures of protein and other macromolecules that are involved in cancer. This sort of structural information is really crucial for understanding how molecules function and interact, which can really help in drug design and drug discovery. And just another example is from our molecular cytology core facility. This is their atomic force microscope. The AFM can measure the stiffness and elasticity of cancer cells compared to normal cells. Cancer cells often exhibit a kind of changed mechanical properties, which can indicate their state of malignancy and invasiveness. So, this can be really crucial in understanding how a tumor is behaving, what its progression is, and also how it's responding to treatment as well. So, really why I believe in digitizing your lab and why I think it's really important is really because this is how you can unlock so much more potential from your lab. You're not tracking down samples, trying to figure out where they are, what aliquots were made. It also ensures you have compliance that you're meeting all of your compliance requirements for your labs, whether you're doing research and you need it to be reproducible, but also for those that are in a translational space as well, making sure that you're compliant with all the regulatory bodies there. And really, because this is how our technologies are evolving, this is how things are moving, and it really just unlocks more potential for all of our labs. So these are just a couple of ways that we have digitized our labs. I want to say on a small scale, but really it's not on a small scale. These are all, while they might seem small or individual things, they are really really meaningful. And then I'm going to kind of go into why, or some real life examples of how these things have facilitated greater research in our core facilities. So first, the electronic lab notebook and lab information management systems that we have. So the ELN is really nice because it allows us to share lab protocols with versioning in a centralized location. And it allows, whether they're contracts that we have with external companies or with other lab members, we're allowed to, or we're able to just share it electronically. So we're not wasting time sharing our physical notes. You're not trying to figure what somebody else has written in their lab notebook. It really just kind of allows that seamless knowledge transfer between different individuals. Um, the LIM system that gives us our predefined workflows to guide the steps of the process, which really ensures our consistency. These can integrate with liquid handling automation that make high throughput processing possible, and it also helps with reproducible data. We have financial tracking, which is really the space that I'm working in right now with the core facilities. It allows the cores to know what it is that they're buying and how often, so that way they can make much more informed decisions. And when you have good financial tracking in your cores, it really poises our core directors and our research labs to make their labs, sustainable and really put them at the forefront of cancer research technologies. We also have utilized sample inventory tracking systems. This I could spend an entire presentation on inventory and sample tracking. um, Knowing the precise location of your samples just saves so much time, energy, and money, really. Instead of spending hours, going through the freezer trying to figure out what you need, whether it's, an Eppendorf that's somewhere in the back of the freezer, or you're trying to figure out, what this label is that somebody, hand wrote on it. Having an inventory and sample tracking system allows you to know where everything is so you can get to it much more efficiently, but also having, printed out labels that are scannable. You don't have to guess anymore what a tube says. And then also, reagent tracking has been really important for us. Really the wasted time and money that is spent in ordering reagents, it really is kind of incredible if you take some time to sit down and think about it. So whether you have, reagents that aren't ordered soon enough and it's going to delay your project, or you've ordered way too much and then they go bad and you end up having to throw them out because they're no longer usable. Um, Embracing digital lab platforms in all of these different ways have allowed us to really see a lot of the potential that all of our core facilities and beyond have had. And so now I'm going to share a couple of nice examples of, how we've done it um, in our real lab space. So first, I'm going to just give a more basic research example on spatial transcriptomics. So first, this slide is just to kind of illustrate what that is. So single cell sequencing has been a really popular technique that can provide you with RNA expression information from individual cells. So one single cell, what is the expression information? And that's really incredibly powerful for understanding how a mutation is changing individual cell expressions of their proteins. However, that information is only so powerful when we consider the 3D landscape that solid tumors are growing in. So, to just kind of illustrate this, imagine that you're opening a coffee shop and you are an entrepreneur and you want to open up a new coffee shop on the Upper East Side of New York City. So, we have a couple of different storefronts. You go online and you take a look at what the different storefronts are like. You might prefer this one because of, the you like the countertops that they have, or you might like this one because you like the lighting setup that it has. But this information really is only so useful. Instead, what you kind of need to think about is really where those coffee shops are. So we have our Upper East Side, and these are where all of our storefronts are actually located. So even though you might have preferred the lighting application in the one in the top right corner, you'll realize that it's surrounded by three other coffee shops. So maybe that storefront isn't the greatest location for you to open your new business. And really, this kind of additional layer of information is what transcriptomics offers to us, just more information, which is particularly useful in our solid tumor landscape. So as you can imagine, this kind of level of information or work is really intricate and requires highly specialized technicians across multiple disciplines. So that means that the samples and the information has to flow between various different cores in order to get the best quality data possible. So really what our digital labs have allowed us to do is have much more collaboration and really be able to perform something as highly intricate as spatial transcriptomics, because we are able to transfer information from one lab to the next, or even to our innovation labs. So in spatial transcriptomics, we have our tissue, which is collected by the investigator. Sometimes it's coming from our pathology core facility, but it's passed off to our molecular cytology core facility. In the molecular cytology core facility, the tissue has to be fixated and the tests of permeabilization have to take place. And these are techniques using different equipment that our molecular cytology core already has, but they're also experts in this kind of tissue manipulation. Then the sample is brought over to our integrated genomics operation core facility, and this is where the library preparation and sequencing happens. Our integrated genomics operation core is really dedicated to all forms of genetic sequencing. And in this case, they already have all of the equipment that's necessary to perform transcriptomic sequencing. Once they've performed all of the necessary steps and actually put the sample into the sequencer, the data itself can then be transferred um, over to our bioinformatics core facility for analysis of the data as well as figure generations for papers. The sample information and metadata is collected and transferred from each core facility to the next, and it's all done electronically. The cytology images themselves are sometimes so big that you don't really want to use up excess memory by having them saved in multiple locations. So we also have all of that data saved in a unified location, so whoever needs to access it can. This also, because all of the information is tracked digitally, it also allows our investigators to be able to see what step in the process their project is at, which, you know, when you're waiting on some samples that you've worked on, or, it's a really interesting tumor type that, you really need some information to drive what are the next steps in your project. This can be, just really nice knowing where things are at. And as more omics platforms are being developed, we're able to integrate different cores into the different parts of the process because they all have this digital lab platform, these digital lab solutions that allow us to transfer information back and forth. So now I'm going to pivot to a bit more of a translational case with our radiopharmaceutical and molecular imaging probes. And really, this is just kind of to illuminate how the work that's happening in core facilities and our research development and innovation groups can move into the clinic. So in I'll call them the RMIP core, they investigate radioactive probes with the goal of finding those that can be used in clinical applications. These agents are used with a molecular imaging procedure called the positron emission tomography scan, also known as a PET scan. And these radio tracers, a lot of them are they are prescription now, so they're being used in patients. So in this case, what our core facility has the capacity to do is to receive these radioactive diagnostic agents, prepare them, and then make them ready for use for diagnostic testing. So in this case, the radio tracer that's specific for proteins that are found abundantly on the surface of prostate cancer cells that is prepared in the RMIP core facility. Then the agent actually binds to, it's injected into the patient and then it binds to these antigens on the prostate cancer cells. And then when the patient goes through the PET scan in the middle, it actually the binding of the antigen and the protein actually it illuminates in where the prostate cancer is in the body, and if it's spread to other parts in the body. So if you can take a look at the picture on the right hand side, in this case you can see there's still disease in the prostate, but there's also traces of it in the vertebra and the ilium as well. So this core facility works with investigators to create new radio labeled tracers and see what information can be deduced from each. The iterations of these tracers has to be very carefully tracked so successful agents can then be passed on to clinical trials. And once they're approved, these doses can be requested electronically from our core facility and then tracked into patients, which is example of what I was just showing here. And finally, I'm going to end on another really exciting translational case with our cell therapy and cell engineering core facility. So, cell therapies are a promising new type of cancer treatment really aimed at boosting the immune system to give immune cells the information they need to better target the tumor cells as foreign and then attack them. So, these types of therapies are used to treat a few types of cancers, particularly blood cancers. And so in CAR T therapies, and sorry, CAR stands for chimeric antigen receptor, and they're performed, or I should say, manufactured in T cells. So the way that this kind of works is it starts with the patient, and they come into the clinic where the doctors will then collect their blood, specifically collecting the T cells from the blood. Those are isolated. Then in our core facility, the T cells are genetically modified to produce a special structure, that's the CAR, on the surface. And the CARs really work to help the T cells recognize and then attach to the cancer cells. The next step is the expansion of this modified T cell population. And then once it's grown into a large enough number, we have enough T cells, they're infused back into the patient. And then the idea is that the CAR T cell seeks out and destroys the cancer cells. So the T cell modification and expansion is all that's happening in our core facility. So on the left-hand side, you can see the picture of one of our technicians all gowned up in the process of performing the manufacturing of these T cells. These CAR productions can only be performed in labs that are specifically rated for clinical use. This level of production requires reagent tracking, lot tracking, instrument usage tracking, environmental tracking. All of this information has to be saved in batch records. These batch records it's the comprehensive set of documents that outlines all of the aspects of this manufacturing process that's taking place for each individual batch. It includes in details about, all of the reagents that have gone in, what equipment, what personnel, what were the results. Everything all has to be contained in these batch records. So as you can imagine, they are hefty files. So this is an example of how those batch records actually kind of look. Each one of those brown folders is for one production. So, historically and even currently in many spaces, this information is all kept in a physical form, and it has to pass physically from group to group to, pass the information on, but also those signatures, which are required for the compliance. So, as you can imagine, it takes a lot of time and effort, not to mention you have to physically track down people. We're in the process of transferring this information into a digital format. So first, we've been really working on the reagent tracking, the inventory tracking, because as I mentioned, for these manufacturing, you can only use one lot number for the whole process. So it's really important that we're keeping track of exactly what it is that we're using in each manufacturing. So we have, currently that's what we are working on. So we have our bill of materials that's all being tracked and, sort of written down for these GMP regulations that we operate under. Um, so we also have the environmental monitoring information. So the air quality, the temperature, and whatnot all needs to be collected and stored electronically. We also have, as was mentioned before, with the signing off, the protocols that are used, everything that's done, everybody who's there, and in part of the process, all of that information is now being put in a digital format so that way it can pass much more seamlessly from group to group. And this us really allows us not only just to collect information, but again to go back and refer to the information. As you can imagine, if somebody came and we had to track down, the information that happened in manufacturing in one of those folders up there, it would take a lot more time than if I'm able to just search it on my electronic lab notebook. And this really ensures patient safety and allows us to react much faster. And I'm just going to end on a quick snapshot of our core facilities. While our primary focus is really to drive research here at MSK, we are open to working with internal investigators. So if you're interested in what services our core facilities have to offer, please let us know. I'm really hoping that I've convinced you why it's useful to embrace digital lab platforms and how it can really help your research. And if you're interested in learning more, I am always happy to chat. But with that, I am going to pass this back to Z.
All right. Let's see. I guess we have another poll question over here. Would you be interested in scheduling a 15-minute introductory call with any of our panelists? So this is myself, Twan, or Marisa from MSK. Would be more than happy to hop on a call and discuss any of the topics that we have discussed today in this webinar. And with that, as you fill out this poll, we'll stop here and pass it on back to Israel from LabRoots to close this up. Thank you very much.
Awesome. Well, thank you so much, panel. We are going to dive into just a couple questions that we have. So if our panelists could jump back on camera, let's tackle some of these questions here. This first one looks like it's for Marisa. So Marisa, if we are a biotech company getting ready for clinical work, how can we take advantage of those core facility services?
That's a fantastic question. So as I mentioned before, our core facilities really are focused on driving the research at MSK, but oftentimes we do have the capacity to work with groups outside of MSK, especially, some of our more specialized core facilities. So, really, it can just start with a conversation. On MSK's website, our core facilities are all listed. We have the contact information all there. You're more than welcome to reach out to us, and we would be happy to chat and just start with just discovery, what it is that you're interested in, what it is that you're working on, and just seeing sort of where our cores can jump in and help you out, and see what the future holds for cancer research and see how we can help.
Perfect. Thank you for that. We do only have time for one last question, and this is for Zareh. What is the #1 hurdle for implementing a digital lab platform like an ELN/LIMS in a new lab?
Yeah, well, this is a great question, and the answer is something that people do not want to hear. And the biggest hurdle are the people that are going to implement it, unfortunately, because you can have the greatest digital solution that's out there, but if there isn't that cultural shift, that mindset shift there within the organization to make that change, regardless how much you try, you will not be able to make that change. And this is where people like Twan, who are specialists in lab digitization, myself and our colleagues, and many other people from other organizations, it's important to align with those folks and be able to put your short-term and long-term goals in front of them, and then work as a team, work as an extension of their team to create that shift from day to day, week to week, and month to month, with constant check-ins and making sure that that stickiness is reached during this transformation. So the biggest hurdle is the people, but also that's also the biggest solution there. As long as people are aligned and they work together, it is possible. You just need to have the right templates and the right kind of day-to-day operational project management skills to be able to do it. And of course, people like Twan and myself are there to assist those labs to do that.
Maybe also to quickly build upon that, I have now seen hundreds of labs go through this journey, and it takes time, and there is a lot of time investment at first. But from every one of them, I hear that they would have done it much sooner if they are seeing the results at the moment, and it's a great feedback to hear. But please take this up and also think about it. Would you do it sooner if you know the results and if you know what you can get out of it?
All right. Well, thank you all so much for your time today, for your important research. Now, to our audience, questions that we didn't have time for and those submitted during the on-demand period will be addressed via the contact information provided at the time of registration. This webcast can be viewed on demand for 1 year until November 13th, 2025. Thanks again to our audience for joining us today, and also to our wonderful and magnificent panelists. Until next time, goodbye, everybody.
Bye.
Thank you. Goodbye.